So in the last video, we talked about how the main purpose of glycolysis is to destabilize sugar, to get the ball rolling, to start releasing energy. And yes, you get out a net 2 ATP. You also load up 2 energy carriers with high energy electrons stripped from sugar or whatever food molecule is going through the process. So let's take a look at this process more carefully. So like I said, you don't need to memorize every step, but I want to display these steps sort of in this simple diagram just to give you an idea of what's going on. So glucose is a 6-carbon sugar. And again, this is not the only sugar or food molecule that goes through glycolysis. It's just the easiest to track because glucose is the molecule that goes through cleanly from beginning to end. So other kinds of energy molecules would come in at different stages. They would need modifications. They wouldn't go through exactly the same way, but the same idea would hold true regardless. So glucose is a 6-carbon sugar, and it is a ring, but in this diagram it's just listed as 6 carbons in a chain. Just these black circles are supposed to be carbons because we're just going to track how many carbons there are, that's all. So you can see here that ATP is added and it releases its energy to the glucose molecule to destabilize it by adding one of those phosphate groups. And we talked about phosphorylation in one of the videos. This is called substrate level phosphorylation because it's one molecule that has a phosphate group just transferring its phosphate to another molecule. So that's really simple version of phosphorylation. It's really easy. You just have a high energy molecule with a phosphate group adds its energy to another molecule by adding the phosphate group. Because remember, those phosphate groups have a lot of electrons packed together and they're all repelling each other. So that's really unstable and high energy. So now this was adenosine triphosphate. It had 3 phosphate groups. It adds its phosphate to sugar and then it leaves as adenosine diphosphate, which is a lower energy version of the same molecule with only 2 phosphate groups leaving. So this happens a second time. ATP is invested. It invests its energy in the form of a phosphate group, a high energy unstable phosphate group, added to the sugar, and then it leaves as ADP. So this ADP, it has to be, so again, it's sort of like ATP is like a rechargeable battery. So it can be charged in the form of ATP, or it can be depleted in energy in the form of ADP, or even AMP, which is adenosine monophosphate, if a second phosphate group is broken off, leaving only one of the 3 left. So those are just energy depleted versions of the same molecule, but they get recharged by adding the phosphate groups back on. But to add the phosphate group back on, you have to invest energy because you're creating a high energy molecule to store that energy. And then you can release the energy by breaking them off. So 2 ATPs were invested. Now it's, they are lost in a way. They're low energy molecules now. But now this sugar molecule is super unstable. It has 2 high energy phosphate groups attached to it. And so now it's super easy to break in half. Now it's unstable enough to start breaking it down and releasing all the energy it contains. So here it's broken in half into 2 different 3-carbon molecules. So just like energy, matter is not created or destroyed. You have the same elements starting as you have finishing. They're just in a different pattern or a different form. So you have 6 carbons here. You break them in half into 2 different 3-carbon molecules. 2 times 3 is 6. So you still have 6 carbons total. And then you start stripping some electrons off these molecules, and you start taking these phosphate groups and adding them back onto ATP. Right? So you add these phosphate groups back onto ATP, and then so I know that there's some pieces of this diagram that we aren't covering because I don't want to make this overly complicated. I just want you to get the main point, which is 2 ATP are invested to destabilize sugar. 4 ATP are gotten out. So these low energy molecules are recharged as ATP. So 2 are depleted, 4 are charged or created, and so that's a net gain of 2. Right? Net gain of 2 ATP per glucose. And then you also have these places where these electron carriers are loaded up with high energy electrons. So NAD+, 2 NAD+ become 2 NADH, the electron heavy form, and these high energy electrons are destined for the electron transport chain where they will be applied to generating ATP, more ATP. So you don't get a lot of ATP, you don't strip very many electrons. The main point here is investing energy to destabilize sugar and then breaking it down and harvesting, yes, a little energy is harvested, but that's just not the main purpose here. All right, so pyruvate. So these three, you'll notice at the end of glycolysis, you finish with two different 3-carbon molecules. And this is a highly simplified image, right? Because these, you can see that this, when you're just tracking the carbons, you're not seeing exactly how these molecules are changing all the way through. And you're just, you've got the carbons in black, the phosphate groups in red. You're not really seeing all of the minor changes that take place from one step to another. But what you have at the end, it says here pyruvate. Pyruvate is a 3-carbon molecule. So you had a 6-carbon molecule to start, and you finish glycolysis with 2 different 3-carbon molecules. And these 2 different 3-carbon molecules called pyruvate are going to be what goes through the citric acid cycle or the Krebs cycle, right, whichever your current textbook is calling it. So pyruvate can continue through the Krebs cycle and the electron transport chain, or it can be taken out and used to synthesize molecules that the cell needs to function and build cell parts, like fats for the plasma membrane, like lipids for the plasma membrane, or amino acids for enzymes and other proteins, or sugars to build carbohydrate structures, like for the cell wall for peptidoglycan or the extracellular matrix. So all the different building, so pyruvate, here you have glycolysis, you put glucose in, pyruvic acid or pyruvate comes out, and this can either go through the Krebs cycle, citric acid cycle, or and continue through the electron transport chain, or it can diverge here and go to making amino acid, sugars, and fats, right? So this, when pyruvate diverges here, it's to build cell parts, because when we talked about chemoheterotrophs, you are chemoheterotroph, the microbes that inhabit your body are chemoheterotrophs. They're not just getting energy from sugars and other molecules that go in, they're also getting building blocks. So yes, we are focusing in this chapter on the energy extraction process, but you should also know that there is a branch here where the breakdown of sugar can go towards building these new cell components so that the cell can build itself, repair itself, make offspring, things like that. All right, so we're not going to go into, I know that there is pyruvate oxidation. So I told you that I lumped them together. So here's the citric acid cycle or the Krebs cycle, and here's pyruvate oxidation. I just lumped them together into one step, and I'll usually refer to them collectively as the citric acid cycle or the Krebs cycle, because they're really to the same end. The overarching idea of those 2 steps are really the same. So the main points are remnants of sugar, which is, it's pyruvate now that's entering. You can see that pyruvate is entering here. So that's a remnant of sugar, is stripped of remaining electrons, breaking the bonds between the carbon atoms and releasing carbon dioxide. So you know that you breathe in oxygen, and you need that oxygen to live, and then you exhale carbon dioxide. And a lot of people then, because of that process where it seems like you're exchanging oxygen for carbon dioxide, so people think that the oxygen they breathe in gets used and converted to carbon dioxide, which they exhale, but that's not true. The carbon dioxide you exhale is actually your food. You break down your food and you exhale a chunk of it as carbon dioxide, right? So here you have pyruvate, which is the remnants from glycolysis, so that's what's left over of glucose from glycolysis, comes in. And there's, these many steps that go round and round and round. It's a cycle. So, these end products from glycolysis come in, they attach to the end products of the last citric acid cycle, and they all go through again. So carbon atoms will actually go through this cycle several times before they get released as carbon dioxide. And again, that's really complicated. We're not going to worry about the minute details of that. But the main point is, you can see, so pyruvate is a 3-carbon sugar. So pyruvate is a 3-carbon molecule that comes in. Here you see carbon dioxide leaves, and you see that that's coupled with this electron carrier getting loaded up with high energy electrons. So NAD plus strips some electrons, becomes NADH. And those high energy electrons are going to go towards generating ATP in the electron transport chain. So the reason stripping electrons is going to break this molecule down is because the 3-carbon molecule is held together by covalent bonds. Covalent bonds are shared electrons. So the atoms are only held together because there are these electrons that orbit all 3 atoms, and the orbiting electrons holds them together. So if you take those shared electrons away, the molecule is going to disintegrate. It's going to fall apart. So you strip these electrons, and one of these carbons gets released as carbon dioxide. So you have a 2-carbon molecule going in. And there's another place where carbon dioxide gets released over here when more electrons are stripped. And there's another place where, let's see, where's the other carbon dioxide? Oh, here. Another place where carbon dioxide is released over here, and NADH is created because electrons are stripped. So there's 3, you come in with a 3-carbon molecule and you release 3 carbon dioxides in this process. And even more electrons are stripped also. So here's another place where NADH is created by stripping high energy electrons from sugar. It says Q here. Q is the same thing as FAD. So this is FAD this is FAD, which becomes FADH2. So that happens here. So remember, the main purpose of the Krebs cycle is to oxidize your food stuff. So you're stripping electrons, stripping those high energy electrons out of the high energy food molecules, and carbon dioxide gets released as a byproduct because of the breakdown of those covalent bonds. So you do generate ATP here, but not that much. So this Krebs cycle shown in the slide is for 1 pyruvate. But remember, you have to double it per glucose, because 2 glucose went into glycolysis, 1 glucose went into glycolysis, 2 pyruvate came out. So 2 pyruvate equal 1 glucose. Okay, so you have to double all of these numbers to get what happens per sugar molecule. So you get 4 NADHs. That means 4 per pyruvate, 8 per glucose. 1 FADH2 per pyruvate, that means 2 FADH2 per glucose. 3 carbon dioxides are released per pyruvate, that means 6 carbon dioxides are released per glucose, which means all the carbons from glucose are released as carbon dioxide in this phase, right? Because glucose was a 6-carbon molecule, and you've released all of those carbons now as carbon dioxide. So this is where the carbon dioxide waste comes from, and I want you to remember that for your quiz. And this is where most of the electrons are stripped, and those electrons are going to drive ATP synthesis in the next step. So before we go over the electron transport chain, which is the hardest part of this, I just want to do a quick review. So energy is harvested by stripping electrons from high energy food molecules like sugar. Electron carriers like NADH and FADH2 carry those high energy electrons from sugar or fats or whatever food molecule it is to the electron transport chain to drive ATP production. That's where most of the ATP is going to be produced. ATP is the energy currency of the cell. ATP is going to carry the energy that was originally bound up in food from the site of energy harvest, where ATP was generated, to the site of the work that needs to be done in the cell. So whether it's moving the flagella so that the cell can move around, whether it's cell division to create progeny, whether it's moving something against its concentration gradient across the cell membrane, it doesn't really matter, but any activity that needs to be done in the cell, ATP is going to drive that. In glycolysis, 2 ATP were invested, 4 ATP were harvested, which means a net gain of 2 ATP per glucose. You also have a net gain of 2 NADH per glucose. And I'll remind you that the main point of glycolysis is not the ATP, it's not the NADH, it's the destabilizing sugar. But you happen to be able to get a few high energy electrons out of it and a few ATP out of it. Second step, Krebs cycle, or the citric acid cycle. All the carbon from the sugar is lost as carbon dioxide, which you will then exhale. 2 ATP are generated, so not that much. But the main purpose of this is loading up those electron carriers with high energy electrons. So 8 NAD pluses are loaded up with electrons to become 8 NADH. 2 FADs are loaded up with electrons to become 2 FADH2s. Then all these electron carriers, all the NADHs and FADH2s from glycolysis and pyruvate and the Krebs cycle are going to go to the electron transport chain. And that's what we'll cover next. In the electron transport chain, all NADH and FADH2 are used to produce somewhere between 28 or 30 to 34 ATP per glucose. The reason this is a range is because it's really hard. Part of the reason is it's really hard to estimate, and that will make sense to you later. But there's a lot of components that happen where there's hydrogen ions being pumped across membranes. It's hard to know exactly how many are pumped to drive exactly how many ATP, but it also varies by species. So prokaryotes and eukaryotes have a slightly different ATP yield per glucose. So here I'm going to give a brief overview of the electron transport chain. In the next video, I'm going to go into a lot of detail. So in the electron transport chain, electron donors, like NADH and FADH2, pass those high energy electrons that they stripped from food to an enzyme complex. Right? There they are. So here are these enzyme complexes embedded in this membrane. Right? And NADH is loaded up with high energy electrons from glucose or other foodstuff. FADH2 is loaded up with electrons from glucose and other foodstuff. So they're going to pass their high energy electrons now to an enzyme complex that's embedded in this membrane. Electrons get passed along a chain of enzyme complexes, and you can see that happening in this animation here. Each time the electron is passed, it loses energy. It's moving to a more and more stable state every time it's passed, which means it's releasing more and more energy as it goes. The energy is used to, the energy that it releases, that electron releases, is used to generate ATP. So let me go through those animations one more time before we go into the high detailed version here. So electron donors, NADH and FADH2, pass their high energy electrons to an enzyme complex. Electrons get passed along a chain of enzyme complexes, and each time the electron is passed, it loses energy because it's always getting moved to a more and more stable state, and there's that inverse relationship between energy and stability. The energy that's released by those electrons is used to generate ATP. So every time the electrons are passed from one enzyme complex to another, they're releasing this energy to generate ATP, but how ATP is generated from that energy is really quite an elegant process. So in the next video, we'll look in more detail about how that happens.